Trillions of microbes ready to invade our bodies lurk in the air, the water, the foods we eat, and on the objects we touch. Nevertheless, most of us are healthy most of the time and unaware of the constant battle between body and microbe. Even when infections occur, we almost always fend them off quickly. Our usual state of good health is due to three lines of defense: external barriers that keep microbes out of the body, non-specific internal defenses that combat all invading microbes, and our immune system, which assaults specific microbes. The body's first defense against microbes are the skin and the mucous membranes of the digestive and respiratory tracts. The dry, dead cells, filled with hard proteins that cover the outer surface of the skin, prevent most microbes landing on the skin from obtaining needed water and nutrients, while acids and natural antibiotics secreted by the sweat and sebaceous glands actively inhibit the growth of bacteria and fungi on the skin. The membranes of the digestive and respiratory tracts are protected by mucus, which contains antibacterial enzymes that destroy the cell walls of bacteria and physically traps microbes that enter the body through the nose or mouth. Cilia on the membranes sweep up the mucus, microbes and all, until it is either coughed up, sneezed, or swallowed. Swallowed microbes enter the stomach, where they encounter a combination of extreme acidity and protein-digesting enzymes that kills many microbes. Farther along the digestive tract, bacteria harmless to humans secrete substances deadly to other invading bacteria or fungi. Though these defenses are good, the warm, moist mucous membranes are more vulnerable to invasion than the skin. Disease organisms that enter the body through the skin or mucous membranes run into three nonspecific internal defenses: standing armies of phagocytic and natural killer cells, the inflammatory response, and fever. Monocytes and neutrophils are phagocytic white blood cells that travel through capillaries to wounds where bacteria have gained entry, then ooze out through narrow openings in the capillary walls. After leaving the capillaries, monocytes differentiate into macrophages, amoeba-like cells that engulf and ingest bacteria and other foreign particles. Neutrophils, the most numerous of white blood cells, like macrophages, also engulf foreign invaders such as bacteria and certain fungi. Both monocytes and neutrophils secrete molecules that attract other defensive cells to the invasion site. Natural killer cells are another class of white blood cells. They do not directly attack and engulf invading microbes, but rather identify and destroy body cells that have been invaded by viruses or that have become cancerous. Sensing molecular changes, natural killer cells bind to the compromised cell's cellular membrane and release proteins that pierce holes in it. Cytotoxic proteins then ensure the destruction of the abnormal cell by entering it and destroying its nucleus. Breaches of the skin or mucous membranes elicit an inflammatory response. Damaged cells release the chemical histamine into the wounded area. Histamine makes capillary walls leaky. Leaky capillaries cause fluid to seep from the capillaries into the tissues around the wound. The wound becomes red, swollen, and warm. Meanwhile, other chemicals released by injured cells initiate blood clotting that both blocks damaged blood vessels, preventing microbes from escaping into the bloodstream, and seals off the wound from the outside world, limiting the entry of more microbes. Still other chemicals, some released by wounded cells and others produced by microbial invaders, attract macrophages, neutrophils, and other phagocytic cells to the wound, where they engulf microbes, dirt, and tissue debris. The pus that collects around a wound consists largely of microbes, tissue debris, and living and dead white blood cells. If enough microbes survive the inflammatory response and infect larger areas of the body, a fever may be triggered. While severe fevers are dangerous, even fatal, average fevers of 38 or 39 degrees Celsius, 100 to 102 degrees Fahrenheit, are beneficial. In the brain, the hypothalamus contains temperature-sensing nerve cells that normally set the body's thermostat at about 37 degrees Celsius or 98.6 degrees Fahrenheit. However, certain white blood cells, in response to infection, release hormones called endogenous pyrogens that cause the hypothalamus to trigger behaviors that increase body temperature, shivering, increased fat metabolism, and a feeling of cold that causes a search for heat and warmth. The release of pyrogens also results in reduced iron concentrations in the blood, which, combined with the fever, slows the reproduction rate of many bacteria. Further, fever increases the activity of the phagocytic white blood cells that attack bacteria. Fever fights viral infections by increasing the production of the protein interferon, which is synthesized by certain body cells when they are invaded by viruses. Interferon released by these cells travels to other cells and increases their resistance to the viral attack. While often effective, these nonspecific defenses are not impregnable. When they fail to do the job, the body then mounts a highly specific immune response directed against the particular organism that has successfully invaded the body. More than 2,000 years ago, the Greek historian Thucydides recognized that occasionally someone contracts a disease, recovers, and never catches that particular disease again. The person becomes immune. With rare exceptions, however, immunity to one disease confers no protection against other diseases. Thus, the immune system attacks one type of microbe, overcomes it, and provides future protection against that microbe, but no others. This is why we refer to the immune response as a specific defense against invasion. The immune system consists of about 2 trillion specialized white blood cells called lymphocytes. Many are distributed throughout the body in the blood and lymph. Others are clustered in specific organs, particularly the thymus, lymph nodes, and spleen. The immune response results from the interactions among several types of lymphocytes and the molecules they produce. The immune response is provided chiefly by 2 types of lymphocytes, B cells and T cells. As with all white blood cells, B and T lymphocytes arise from cells in the bone marrow. Early in development, newly forming T cells migrate to the thymus, hence the name T cell, and differentiate. B cells differentiate in the bone marrow itself, hence the name B cell, and later migrate typically either to the spleen or lymph nodes. Immune responses produced by both B and T cells consist of 3 steps: recognizing invaders, launching an attack, and retaining a memory of the invader to ward off future infections. To understand how the immune system responds to invading microbes, 3 questions must be answered. How do immune cells recognize foreign molecules? How do they produce specific responses to so many different molecules? And how do they determine that a substance is foreign and not self, thereby avoiding destruction of their own body tissues? The key to the immune system's ability to attack invading microbes lies largely with proteins called antibodies and T cell receptors. Antibodies are either attached to the surfaces of B cells or dissolved in the blood plasma, where they are called immunoglobulins, often abbreviated IG. T cell receptors are only found attached to the surfaces of T cells. Antibodies are Y-shaped molecules composed of 4 peptide chains, 1 large, heavy chain, and 1 small, light chain on each side of the Y. Both heavy and light chains consist of a constant region that is similar in all antibodies of the same class and a variable region that differs among antibodies. The combination of light and heavy chains results in an antibody with 2 functional parts, the arms, and the stem of the Y. The variable regions on the arms form highly specific binding sites for large molecules. Each binding site has a particular shape and electrical charge, so only a few, or perhaps only one type of molecule can fit in and bind to the site. In general, only large, complex molecules, such as proteins, polysaccharides, and glycoproteins, bind to antibodies. These molecules, or antigens, may either be attached to the surfaces of cells, the body's own or invading microbes, or dissolved in the blood or extracellular fluid, such as snake venom. As we will discuss shortly, the binding of antigen to antibody triggers the immune response. The stem of antibody molecules, which consists of part of the constant regions of the two heavy chains, determines the activity of an antibody. Though the constant regions in different antibodies are similar, they are not identical. For example, the heavy chain constant regions forming the stem of one type of antibody may attach the antibody to the plasma membrane of a cell. The constant regions of another type of antibody may bind to certain proteins in the blood, called complement, to promote the destruction of microbes.